In last video, we have discussed about medicinal use of microorganisms and discoveries of vaccines by different scientists and increasing soil fertility, harmful microorganisms and disease causing microorganisms in human. Next we will see disease causing microorganisms in plants. Several microorganisms cause diseases in plants like wheat, rice, potato, sugarcane, orange, apple, and others. The diseases reduce the yield of the crop. If these plants are affected by disease, it reduces the yield of the crops. They can be controlled by the use of certain chemicals which kill the microbes. Here chemicals are nothing but the pesticides. Plant pathogens are very similar to those that cause disease in humans and animals, like fungi, fungal-like organisms, bacteria, phytoplasmas. Phytoplasmas are bacterial parasite of plant. Viruses, viroids, and nematodes. Viroid is a plant pathogen and nematodes are the worms. Next we will discuss about common plant diseases caused by microorganisms. First one is citrus canker. Causative microorganism is bacteria. Mode of transmission through air. Next, rust of wheat. Causative microorganism fungi. Mode of transmission through air or seed. Last disease is yellow vein mosaic of bhindi. It's also called as okra. Causative microorganism virus. Mode of transmission insect. Citrus canker is a disease affecting citrus species like lemon, orange, etc. caused by the bacterium Xanthomonas. Rust of wheat, it is the fungal disease that affect wheat and barley grains. Next, yellow vein mosaic of bhindi or okra. It is a viral disease. Now so far we have discussed totally 3 diseases: citrus canker, rust of wheat, yellow vein mosaic of bhindi. These are the common plant disease caused by the microorganisms. Next we will discuss the chemical method to prevent the attack of microbes. Salts and edible oils are the common chemicals generally used to check the growth of microorganisms. Therefore, they are called preservatives. Preservative is a substance or chemical that is added to products such as food product, etc. to prevent decomposition by microbial growth. We add salt or acid preservatives to pickles to prevent the attack of microbes. Sodium benzoate and sodium metabisulfite are common preservatives. They are also used in the jams and squashes to check their spoilage. Here squashes are the fruit, liquid or juice. Here in chemical method, the preservatives we have discussed, that is salt, edible oils, sodium benzoate and sodium metabisulfite. These are the preservative which can prevent the food from spoiling and from decomposition by microbial growth. First we will see preservation by common salt. Common salt has been used to preserve meat and fish for ages. Means for the longer time we can preserve the meat and fish in the common salt. Next, preservation by oil and vinegar. Use of oil and vinegar prevents spoilage of pickles because bacteria cannot live in such an environment. Vegetables, fruits, fish and meat are often preserved by this method, and this common salt, oil and vinegar is easily available and we can preserve our food and prevent decomposition by microbial growth. Next we will see preservation by sugar. Jams, jellies, and squashes are preserved by sugar. Sugar reduces the moisture content, which inhibits the growth of bacteria which spoil food. Inhibits means prevents. The sugar prevents the growth of bacteria which spoils the food by adding the sugar in that food. Next, heat and cold treatment to preserve the food for the longer time. You must have observed your mother boiling milk before it is stored or used. Boiling kills many microorganisms. Similarly, we keep our food in the refrigerator. Low temperature inhibits the growth of microbes. Whenever we boil the milk or water or any food substance, all the microorganisms will be killed. Similarly, when we keep the food in the refrigerator, because of the low temperature, all the growth of the microbes will be inhibited. By these all chemical methods, we can preserve the food for longer time. Now, once again, we will summarize the microorganisms by looking at the next video.
Variety of plants and animals around us. However, some living organisms are not visible to the unaided eyes. These tiny organisms are called microorganisms or microbes. Note that not all tiny organisms are termed as microbes. Microorganisms are found in air, water, soil, and also inside the bodies of plants and animals. Let us perform an activity to understand this. Take some moist soil in a beaker and add water to it. After the soil particles have settled down, observe a drop of water from the beaker under a microscope. You will find many tiny organisms moving. You can also try this activity using a few drops of water from a pond. Some microorganisms, such as fungi, that grow on moist bread, can be seen with a magnifying glass, while others are microscopic. Hence, they are called microorganisms or microbes. Microorganisms are classified into 4 major groups, that is bacteria, fungi, protozoa, and some algae. Some examples of bacteria are spiral bacteria and rod-shaped bacteria. Those of fungi are bread mold, penicillium, and aspergillus. Amoeba and paramecium are examples of protozoa, while chlamydomonas and spirogyra are examples of algae. Bacteria, some algae, and protozoa are single-celled, whereas fungi and some algae are multicellular. Viruses, though different from these organisms, are considered microbes. They reproduce only inside the cell of the host organism, such as bacterium, plant, or animal.
Next we will discuss about the famous microbiologist Louis Pasteur and pasteurization process. Louis Pasteur was a French biologist, microbiologist and chemist, renowned for his discoveries of the principle of vaccination. Renowned is nothing but the famous. He is famous for his discoveries of principle of vaccination and microbial fermentation and pasteurization process. He is remembered for his remarkable breakthroughs in the causes and prevention of diseases, and his discoveries have saved many lives ever since. Now next we'll see what is pasteurization process. This Pasteur is best known for inventing the process that bears his name. Pasteurization kills microbes and prevents spoilage in milk and beverages by heating and allowing them to cool. Pasteurized milk can be consumed without boiling as it is free from harmful microbes. Milk is heated to about 700 Celsius for 15 to 30 seconds and then suddenly chilled and stored. By doing so, it prevents the growth of microbes. This process was also discovered by Louis Pasteur. It is called pasteurization. Next, storage and packing. These days, dry fruits and even vegetables are sold in sealed airtight packets to prevent the attack of microbes. The Louis Pasteur is one of the microbiologists. He given the concept of pasteurization, that is nothing but heating the liquid or milk about 700 degrees Celsius for 15 to 30 seconds and then suddenly chilled and stored. That process is called as the pasteurization. Next is the main important concept, nitrogen fixation. You have learned about the bacterium Rhizobium in class 6th and 7th. It is involved in the fixation of nitrogen in leguminous plants. Leguminous plants are nothing but the pulses, nuts, etc. Recall that Rhizobium lives in the root nodules of leguminous plants such as beans and peas, with which it has a symbiotic relationship. Symbiotic relationship is nothing but both the organisms will get benefit. Here the Rhizobium bacteria will get shelter and in return the leguminous plants will get the nutrition from the Rhizobium bacteria. This Rhizobium bacteria lives in the root nodule leguminous plants such as beans and peas, with which it has a symbiotic relationship. Sometimes nitrogen gets fixed through the action of lightning. Earlier we have discussed the nitrogen is introduced to the soil by precipitation or by the lightning process. But you know that the amount of nitrogen in the atmosphere remains constant. You may wonder how. Let us understand this in the next section by nitrogen cycle. Now we'll see how the amount of the nitrogen is constant. Our atmosphere has 78% of nitrogen gas. Nitrogen is one of the essential constituents of all living organisms as part of proteins, chlorophyll, nucleic acids and vitamins. The atmospheric nitrogen cannot be taken directly by plants and animals. Certain bacteria and blue-green algae present in the soil fix nitrogen from the atmosphere and convert into compounds of nitrogen. Once nitrogen is converted into these usable compounds, it can be utilized by plants from the soil through their root system. Nitrogen is then used for the synthesis of plant proteins and other compounds. Animals feeding on plants get these proteins and other nitrogen compounds. When plants and animals die, bacteria and fungi present in the soil convert the nitrogenous waste into nitrogenous compounds to be used by plants again. Certain other bacteria convert some part of them to nitrogen gas, which goes back into the atmosphere. As a result, the percentage of nitrogen in the atmosphere remains more or less constant. Now we learn the nitrogen cycle in cyclic form. The first one is here atmospheric nitrogen. The nitrogen is introduced to the soil by precipitation process or lightning. Lightning fixes nitrogen. Then The bacteria in the soil and the plant roots also convert nitrogen into usable forms. That is nothing but compounds of nitrogen in the soil. And in that soil, the usable plants will grow, uptake of plants, and that plants will be eaten by the animals. The animals will get that nitrogenous compounds, and the waste of that animals means excretion that goes again to the soil. From that soil, bacteria turns compounds of nitrogen into gaseous nitrogen. Now in detail, we will learn the nitrogen cycle by the video.
Our last cycle that we'll be learning about is the nitrogen cycle. Nitrogen is really important to both plants and animals and all living things on this planet. Please follow along with me as I go through the notes. Feel free to take notes as I talk and also write down each step of the nitrogen cycle that correlates with the diagram. At step 1, nitrogen is introduced to the soil by precipitation. Nitrogen exists in our atmosphere naturally. 78% of our atmosphere is made up of nitrogen. The other 22% being made up of oxygen, carbon dioxide, and other greenhouse gases. As rain falls to the ground, it brings elemental nitrogen from the air down into the soil, which brings us to step number 2. At step number 2, bacteria in the soil and in plant roots change the nitrogen from the air into a more usable form for plants. This is called nitrogen fixation. Now that plants have this usable nitrogen, it brings us to step 3. At step 3, plants build up proteins and DNA using this usable nitrogen they get from the soil. This brings us to step 4. At step 4, animals eat the plants and they use the nitrogen inside the plants to build their own proteins and DNA. Steps 5 and 6 kind of go together. At these steps, waste products and dead organisms decompose. This releases nitrogen back into the soil. Finally, we have step 7. At step 7, bacteria in the soil break down these nitrogen compounds and release nitrogen gas into the atmosphere. And the cycle starts all over again. So to review, 1. Nitrogen is introduced to the soil by precipitation. 2. Bacteria in the soil and plant roots also convert nitrogen into usable forms. This is called nitrogen fixation. 3. Plants build up proteins and DNA using usable nitrogen absorbed from the soil. 4. When animals eat these plants, they use it to build animal protein and DNA. 5 through 6. When these animals poop, pee, or die, the waste products or dead bodies are broken down by decomposers and the nitrogen is reintroduced into the soil. 7. Nitrogen compounds in the soil can also be broken down by bacteria and sent into the air as nitrogen gas. Once the nitrogen gets back into the air, the cycle continues.
The key words we have learned in this chapter: algae, antibiotics, antibodies, bacteria, communicable disease, fermentation, fungi, lactobacillus, microorganisms, nitrogen cycle, nitrogen fixation, pasteurization, pathogen, preservation, protozoa, rhizobium, vaccine, virus, yeast. Now you should remember these points and description. This completes your second chapter.